Hello. We're going to talk about the other two documents for the Human Genome Project. Now, this document was, for these two documents, we're just going to look at it very briefly, just on some of the important points, because the whole idea of this, looking into these documents, is for you to understand the works, the Human Genome Project. So, in the second document that was published in 1993. When this document was published, the Human Genome Project has gone on for at least 2 years and a bit, and they've actually found quite a lot of, they've learned a lot. The project is on track, but they have additional information now, they have more experience, and that they thought that they should relook at the original goals and then set new goals. Some of the things they learned were at that time, by the year 1993, is that now they have new genetic markers. They've identified the microsatellite as the better choice for markers, and these are in abundance, because previously they have relied quite a lot on RFLP, restriction fragment length polymorphism. They've also improved the cloning systems. Remember they started off with YAGs, and then they found that the YAGs were not very stable. So most of the genome projects rely on the use of BACs, bacterial artificial chromosomes. They have refined and increased a lot more of the sequence tag sites, and they agreed that the STSs are the main reference for physical mapping. And then also they have improved on automation of the DNA sequencing. Automation here means they've improved methods to do sequencing in bulk. They have robots that does DNA extraction, and in various stages, also cloning and the works. Now, the second thing that they learned was that initially the focus of the Genome Project was based on individual chromosomes, but with the advent of additional technologies, they were now thinking that perhaps they should look at the genome as a whole. This relies again on the availability of efforts as well, of robotics, as well as new technologies, and they now are looking at whole genome mapping efforts. An important insight into the Human Genome Project is also by the fact that this time around they actually realized that they needed to also do the gene identification in line with the progress of getting the sequences. Now, initially they thought that get the sequence first, and then only then they will look for genes. But with the establishment of ESTs, or express sequence tags, and cDNA libraries, so they thought that no, finding genes should work hand in hand with getting new sequences. So this is what it means here. It was implicit in the original plan. That means it wasn't really stated clearly, but in this time, in the second plan, they said, very specifically, they underlined that they need to look at find new genes, or identify human genes. The rest of the document just talks about what are the goals, specific goals. Some of them, some of the original goal remains. On sequencing, they've managed to cut it down, but they want to do more. They can do it cheaper, but they need technologies that can speed up the sequencing. Here in gene identification, so as I mentioned just now, this was not part of the original goal, so it is now the goal for the second phase. So develop efficient methods in identifying genes and for placement of known genes on physical maps. So this is the ESTs, or sequence DNA. they need to improve. So these are part of the original goals, and they are starting clearly defined targets for model organisms. Some of them have been finished, some of them are close to finishing, and then with that they will develop the informatics. Informatics is like, you know, going back to how to create, develop, how to store, and how to make this information accessible, and then you can do analysis on it. So they need to work on that, because now by this point there's a lot, the amount of data, sequence data, has increased exponentially. On LC, it remains that they need to identify the issues, and so that's about it. On to the next document.